Hi, hello, I'm Sandhya. I'm your botany lecturer. In this lecture, we are going to study about the biomolecules. So, what are biomolecules? The name itself says that these are the molecules or chemical substances which are present in the life or living organisms. So, all the organic compounds comprising of carbon compounds are nothing but the biomolecules. Or you can say all the carbon compounds obtained from the living tissue are nothing but the biomolecules. So all the carbon compounds which are obtained from the living tissue are nothing but the biomolecules, or you can say organic compounds obtained from the living tissue are nothing but the biomolecules. So we cannot say that a single molecule is responsible for the life. It is nothing but it is the interaction of so many molecules. It is the interaction of so many carbon compounds which is making the life possible, which is making the chemical reactions occurring in the life possible. So some of all the metabolic reactions is nothing but metabolism. The metabolism is nothing but sum of all metabolic reactions. So this metabolic reaction is not occurring because of a single molecule, single biomolecule. It is occurring because of the interaction of so many biomolecules. So according to Linus Pauling, life is possible or life is existing because of the interaction of various biomolecules, and it is not the property of a single molecule, but it is the interaction of the various biomolecules and the collection of sum and total of all the different types of the collection and total and sum of all the types of biomolecules, those compounds or ions present in a cell is called as the cellular pool. So what does the scientist says? According to Linus Pauling, life exists due to the interaction of various biomolecules and is not the property of a single molecule, but it is the collection or sum or total of different types of biomolecules, compounds, ions present in a cell, and that is nothing but the cellular pool. So what are biomolecules? You tell me now. What are the general chemical substances or organic compounds or carbon compounds which are present in the living organisms? They are general. What are they? Carbohydrates, proteins, fats. These are the organic compounds or the carbon compounds which are present in the living tissue. And also we can say nucleic acids also. Nucleic acids. So this is the general biomolecules and general biomolecules. And how to analyze them, how to separate them, how to study about them. How can we separate these biomolecules from the living tissue? We are going to study now how to analyze the biomolecules now. How to analyze the carbon compounds. Simple. Take a living tissue, whether it is a leaf or a liver. Take a small tissue of a leaf or a liver. Then you grind it in a mortar or a pestle. Okay, this is the mortar or a pestle. Okay, take that little leaf. If you are taking a leaf or if you are taking a liver, take a small piece, put it here and grind it. And you add trichloroacetic acid. Trichloroacetic acid. Okay, trichloroacetic acid. Wait a minute. Trichloroacetic acid. Okay, when you add trichloroacetic acid, what happens for it? It becomes into CH3COO4, isn't it? Trichloroacetic acid. You add trichloroacetic acid, then what happens? That leaf or a small liver tissue, okay, adds trichloric acid, it will form into a slurry. So a slurry is obtained. Now this slurry, take this slurry and filter it with the help of a cheesecloth or a cotton cloth. Okay, take a funnel, then put here the cheesecloth or the cotton cloth and pour the slurry. So what happens? What happens? Here on the cloth you are going to get the acid insoluble portion. Acid insoluble portion. Then here in the beaker as a filtrate or the liquid, this is the filtrate, isn't it? This is the filtrate. This is the residue on the filter paper or on the cotton cloth or the cheesecloth. The residue is present. Here inside the beaker, the filtrate is collected. This is the liquid. Okay, this is the acid soluble portion. Acid soluble portion. So both in both the cases, in acid soluble portion and acid insoluble portion, the biomolecules are present. The carbon compounds are present. Okay, carbon compounds are present. This is somewhat like, you know, like if you take a milk, add chai patti to it or tea powder to it and boil it, then again you filter it. What happens on the sieve? On the sieve, all the chai patti will be there because it is insoluble. No, that's why it stays on the sieve. It is insoluble. And what all is soluble? The milk and the sugar. What all is there? The water. That all soluble dissolved portion is going to enter into the glass. That is the filtrate. The tea is nothing but the filtrate, where the milk, sugar and water has got mixed. What all is insoluble, that stays on the sieve. The same in that way only. Okay, on the sieve, the acid insoluble portion. Inside the beaker, the filtrate is the acid soluble portion. So in both the cases, they contain the biomolecules. Okay, in both the cases, but they contain the biomolecules. Like this, we can separate the biomolecules. Okay, and here in the acid soluble portion is going to have 18 to 800 daltons of molecular weight substances. Here more than 1000 Dalton molecular weight substances are present in the acid insoluble portion. Molecular weight. This one 18 to 800 Daltons, or you can say less than 1000 Daltons are present. Less than 1000 Dalton molecular weight substances are present in the acid soluble portion. Then like this we can separate out about biomolecules. And also if you take a living tissue, a leaf or a liver piece, and weight, that particular weight is called as wet weight. Sometimes what happens, we are going to wait after drying also. There is all the water evaporates and gets dried. So that particular weight of the living tissue is called as dry weight. Both dry weight and wet weight are important for the living tissues. Like for example, tea powder, whatever we are using, it's a dried leaves. So the fresh leaf weight will be different. The dried leaf weight will be different. So why we have to calculate this wet weight and dry weight? Why? Because it is going to say something about the chemical composition, something about the adulteration. If any adulteration has been done, the purity of it we are going to identify. The purity of it, of the living tissue, of the living thing, of the product, we can identify. Okay, and also ash. What is ash? When you take a leaf and it is burn it. So when you burn it, what happens? The compounds are going to get oxidized to gaseous forms. Gaseous form, carbon dioxide and water vapor, and are removed. So what is the remaining? The remaining one is nothing but the ash. You are going to weigh it. If it is an adulterated one, the weight may be more or less. But if it is a pure one, we are going to have some normal values. When we compare with that of the product, we are going to get the correct information whether it is adulterated or the pure one. So like this, we are going to analyze the living tissues or living organisms. Like what is the chemical composition of them. Why we have to analyze means to know the purity of it, or to get the maximum amount of purity. If we consume the living things consume the living things in the sense, like if you take the clove, ilachi, all these are the plant products. So when we are consuming, when we are weighing, they should be of pure. They should not be adulterated. So to identify the purity of them, to avoid the adulteration, we can take into consideration of these things. Like this, we can analyze the living tissues. Coming on to this portion now. This acid insoluble portion is nothing but it is going to have the substances of molecular weight more than 1000 daltons. Acid soluble portion is going to have the substances less than substances of molecular weight less than 1000 dalton. So what all substances or biomolecules which are present inside the acid soluble are nothing but they are called as biomicromolecules. These molecules are called as biomicromolecules. Acid soluble portion. In acid insoluble portion, biomacromolecules are present. Biomacromolecules are present, okay? Biomacromolecules are present. So, if we talk in terms of chemistry part, these biomolecules or these chemical substances can be divided into aldehydes, ketones, based on the functional group. Aldehydes means CH2 group, ketones means C double bond group, acids means COO group. Then acids like this. Acids means the COO group. Like this, and aromatic compounds. Chemistry point of view, these biomolecules can be divided into like this: aldehydes, ketones, acids, and aromatic compounds. Okay. Based on biological point of view, these biomicromolecules and biomacromolecules can be the examples for them. Biological point of view. Okay, so biomicromolecules will be having sugars, amino acids, fatty acids, nucleotide bases are the examples of biomicromolecules, biological point of view. If you come to the examples of biomacromolecules, the polysaccharides, proteins, nucleic acids, then lipids are the examples for the biomacromolecules. Okay, biomacromolecules. Okay, so if you see about the biomicromolecules, these polysaccharides, proteins, and nucleic acids are all polymeric forms. They're all polymeric forms, except lipids. These nucleic acids, proteins, and polysaccharides are polymeric forms. Lipids are not polymeric forms. So polysaccharides, proteins, they are all polymers. Okay, I'm going to show now. So what are general biomolecules? We said carbohydrates, proteins, fats, nucleic acids. These are general biomolecules, isn't it? So these biomolecules, this particular biomolecules can be divided into two types. Okay, can be divided into biomicromolecules, biomacromolecules. Why these are called as micromolecules? Why? Because they are having molecular weight to 18 to 800 Dalton of molecular weight. The substances are having the molecular weight in between 18 to 800 Dalton or less than 1000. Those substances are present in micromolecules. Here, more than 1000 Dalton molecular weight substances are included in a macromolecules. These are present in acid soluble portion. These are present in acid insoluble portion. Insoluble portion. So, insoluble portion. Then, what are included here? Examples here, biomicromolecules, sugars, amino acids, nucleotide bases, then fatty acids are included in biomicromolecules. Coming on examples here, polysaccharides, proteins, nucleic acids, and lipids. Okay, so in this, these three are polymers. Polysaccharides, proteins, and nucleic acids are polymers, whereas lipids are not polymers. So when we are taking polymers means a group of small substances combined to form into a complex substance. So polysaccharides, proteins, and nucleic acids are complex forms of the simple molecules. Okay, so let's study about those things. Okay, so if you take carbohydrates, so carbohydrates, isn't it? Carbohydrates are nothing but sugars. If you take carbohydrates, they are nothing but having carbon, hydrogen, and water. Okay, they are having carbon, hydrogen, and oxygen, or you can say hydrates of carbon. They're having carbon, hydrogen, and oxygen, or you can say hydrates of carbon. So the simple forms of carbohydrates are what? They are nothing but sugars. Okay, they are nothing but sugars. And this sugars are divided into these sugars when combined. The two sugars are combined. If it is a single one, it is a monosaccharide. If it is again fused into with this monosaccharide, two saccharides are combined, it forms into disaccharide. The disaccharide will form into polysaccharide. So these sugars are micro, whereas these polysaccharides macro. So it is nothing but a polymer. From a simple one, from the combination from the combination of the simple sugars, they are forming into a complex molecule. Polymer of sugars is nothing but polysaccharide. The combination of sugars are going to form into polysaccharide. The simple one is the saccharide, which is a micro. Polysaccharide is a macro. This is about carbohydrates. So if you take proteins again, proteins are generally having the simple ones are nothing but amino acids. So these amino acids will be combining to forming into peptides to polypeptides to proteins. Okay, this are proteins. So amino acids are nothing but the micro ones, whereas the proteins are the macro ones. Okay, if you take fats now, again fats. If you take fats, fatty acids and glycerol. Fatty acids and glycerol are micro. They are simple substances. They combine to form into fats and lipids, which are nothing but they are macro. These are macromolecules. Okay, nucleic acids. Nucleic acids also very nice. The nucleic acid bases. I'll write here. The nucleic acid bases, then nucleosides. Nucleotides are all coming under micro. They form into nucleic acids, which are nothing but macro. Okay, they are polymers. These are polymers of simple substances. So this is about this, and also based on the function also, these biomolecules are divided. Based on the function also, biomolecules are divided. So these carbohydrates, proteins, fats, they all play an important role in the physiological process, metabolism. They play an important role. These are the chemical substances or biomolecules which are going to play an important role in the physiological processes. So those metabolites, those chemical substances are called as primary metabolites. So the substances or the metabolites or the biomolecules which are formed as a part of the metabolism are called as primary metabolites. So all this will be coming out primary metabolites. Then in plants and other living organisms, some metabolites are formed. Some metabolites are formed which do not have any physiological role. They do not have any particular function in the particular living organism. So such type of metabolites are called as secondary metabolites. Secondary metabolites are those metabolites or chemical substances or biomolecules which do not show any identifiable physiological role. They do not have any physiological role inside the body. But ecological importance is there. Very much ecological importance and value is there. If you say examples of secondary metabolites are nothing but rubber, resins, alkaloids, tannins. So all these are the secondary metabolites which are formed as a process of the as a byproducts in the metabolism. These metabolites are formed and they do not have any function, any physiological role. Such type of metabolites are called as secondary metabolites. The examples are rubber, resin, alkaloids, tannins, etc. Many are there, etc. They do not have any physiological role, but they have an important ecological importance and they are useful for the human welfare. They are very much useful for the human beings. Thank you.